In response to the European Space Agency 9th mission call, we propose a mission to monitor the primary productivity of the oceans using our satellite AquaCat, which will measure chlorophyll A and sea surface temperature.
These are the areas we are going to outline in this video.
The aims for our mission are, firstly, to comprehensively measure the chlorophyll A concentration and sea surface temperature in marine environments as an indicator of changes in the temporal and spatial variations in marine primary productivity. Our second aim is to produce a map of global coverage which can be used to indicate the location of plankton blooms with application of how anthropogenic activities can be affected by and are affecting humans.
Our mission responds to the European Space Agency's call for a satellite which will provide innovative solutions to pressing scientific and social challenges. The focus of this call, as laid out in Earth Observation Science Strategies for ESA, is to deliver the capabilities of Earth observation to address the issues facing society today. AquaCat will provide data that will assist in addressing 4 of the 6 social issues put forward by ESA. Issue 1. ESA lays out that over the next 20 years, food production will need to rise 4-fold in order to meet growing demands by a growing global population and improvements in the standards of living. More than 3.5 billion people depend on the ocean for their primary source of food. The ocean's productivity, therefore, is critical to ensuring that society as a whole can continue to be fed into the future. Specific events, such as anoxic conditions caused through hyperproductivity, have caused food shortages throughout coastal regions in Africa, so monitoring these events is essential. Issue 2. Availability of fresh water. In July 2010, the United Nations agreed that access and provision of clean drinking water should be a fundamental human right. Water toxicity limits the access of clean drinking water, and chlorophyll A measurements have been shown to provide a mechanism of early warning of these events. Having an accurate global data set would allow local monitoring of safe drinking water around the world. Issue 3. Climate change. The level of carbon dioxide in our atmosphere is a key contributor to climate change. Levels currently stand at around 400 parts per million, and the IPCC predicts that these will continue to rise through the next century, with potentially devastating consequences. The oceans have absorbed 2/3 of the carbon dioxide emissions in the last century, and primary productivity plays a key role as a carbon sink. Issue 4. The health of the planet and humankind. Marine ecology is driven by primary productivity. This is essential as it provides the first trophic level in virtually all ecological webs. However, excessive primary productivity results in eutrophic conditions and damage to often sensitive ecological balances. Poor water quality not only affects living species in these environments, but also the humans that rely on its output. Death and disease caused by polluted coastal waters cost the global economy.
Our satellite will be measuring chlorophyll A as an indicator of primary productivity. Primary productivity is the measure of biomass. Chlorophyll is the photosynthetic pigment found in phytoplankton. Phytoplankton performs the majority of primary productivity in the ocean. A plankton bloom is defined as a rapid and marked increase in the local population of plankton. This can occur in a matter of days. Main factors that cause blooms to occur are sunlight, nutrients, and changes in water temperature. During winter, plankton activity is at a low level due to lesser amounts of sunshine during winter days and lower water temperature. In spring, an increase in temperature and sunlight is seen. This allows plankton growth and so utilises a high concentration of nutrients in the water through photosynthesis and reproduction, which increases at an explosive rate. This is a problem as explosive growth can rapidly deplete available nutrients, causing a decrease in productivity. This can cause eutrophication, as the concentration of oxygen in a body of water decreases, resulting in fish deaths. This process has a seasonal occurrence at a large scale, such as in the North Atlantic. In spring, the Benguela upwelling occurs off the coast of South Africa. This is one of the most productive areas for phytoplankton in the world. The blooming of phytoplankton is often found along the coast, where there is a higher supply of nutrients. This is often linked to human impact, such as sewage and waste inputs into the ocean. Fertilizers are another cause of phytoplankton blooms, causing environmental devastation. For these reasons, it is important to measure concentration of chlorophyll A to understand causes and effects of phytoplankton blooms. Using satellites to monitor phytoplankton through chlorophyll A and temperature measurements is not a new concept. Missions such as the Coastal Zone Colour Scanner from 1978 to 1995, Marine Observation Satellite, which was launched in 2002, and Sea Viewing Wide Field of View Sensor from 1997 to 2010 are all examples of past and present missions. Our AquaCat mission intends to build further on this data collection.
Our mission will use a sun-synchronized orbit satellite at an altitude of roughly 600 to 800 kilometers. This type of orbit gives almost complete global coverage, good spatial resolution, and allows images to be compared from the same season over several years. In our mission, we intend to use a constellation of satellites to obtain more coverage of Earth per day and allow more frequent observations. This is important for our mission, as phytoplankton blooms can occur over a matter of days, so there is a need to have higher resolution of data and more frequent observations. We will use a multispectral radiometer sensor, which can detect visible light and thermal-infrared radiation being emitted from the Earth's ocean surface. An algorithm for calculating the chlorophyll A concentration requires 3 or more spectral bands, all between wavelengths of 440 to 670 nanometers, by calculating a ratio of blue to green wavelengths. This has been the AquaCat mission proposal. Over and out.